Hello, everyone, and welcome to today's live broadcast, Rapid Variance Interpretation and Reporting using the QIAGEN Knowledgebase and QIAGEN Clinical Insight Interpret, presented by Jennifer Poitras, Ph.D., Principal Genome Scientist, QIAGEN. I am Christina Mahalik of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by QIAGEN. For more information about our sponsor, visit QIAGEN.com. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the button in the bottom left corner and follow the process to obtain your credits. Now, please join me in welcoming Dr. Poitras. I will now turn the presentation over to her.
Great. Thank you so much, Christina, for that introduction and for the opportunity to speak to all of you today. So first, by the way of the agenda, first I'll go through an introduction to the QIAGEN Knowledgebase, then introduce our QIAGEN Clinical Insight Interpret tool, and demonstrate how the knowledge base underlying it enables rapid and accurate variant interpretation. Then I'll show the tool in action with a clinical exome use case where the proband has two unaffected parents and is exhibiting a constellation of features, and through using the knowledge base within QCI Interpret, we were able to rapidly identify and report a pathogenic candidate causal variant. So we're really going to be focusing on the tertiary analysis or on the interpretation portion of this at the end of the pipeline here, but just to put things in context, so the goal really at QIAGEN is to take us from sample to insight. So we do offer a wide variety, and you're likely familiar with our DNA and RNA extraction kits, as well as some library preparation kits to prepare you to perform your next generation sequencing. But once you've generated those FASTQ files, you can easily feed those into our biomedical genomics workbench. And this is a graphical user interface application that's truly a one-stop shop for NGS analysis, empowering the scientist or clinician with really limited bioinformatics knowledge to map reads to the reference and, for the purposes of today's discussion, call variants to produce a VCF file. Which brings me to the topics that I'll discuss today on how we can leverage our curated content within our downstream tools here. Particularly QIAGEN Clinical Insight, which is our clinical decision support platform, to rapidly distill this list of variants down to one or a handful that are likely implicated in the patient's disease. So the end result using this QIAGEN Clinical Insight Interpret tool is a report with relevant variant information, including our automatically classified variants based on the ACMG guidelines, as well as other relevant pieces of information about your variants, including selected citations that are relevant to this variant from the literature and other externally hosted websites. Additionally, you'll see that there's, well, it's written very small here, but I'll cover this later in the talk, a number of data sources that really come together to culminate in the knowledge base that really gives us the power to generate such a report. So accurate, really the crux of this whole thing is that accurate variant interpretation is really no small undertaking. So in order to align with the ACMG guidelines and truly assess the pathogenicity of a variant, several resources are necessary for gathering information. So I've just highlighted a handful here, but we need to sift through variant-specific publications and then go through and look at public and proprietary databases to say, okay, has my variant been seen before in the public domain, and has it been seen in the context of my particular disease context? Additionally, we can look for things like variant frequency or allele frequency in the general population, as well as bring in silico predictors, all to come together to get this variant classification. Now, there are really two main challenges that are faced here. One is, the challenge of efficiency, because first of all, you're going through and looking at all of these different pieces of information independently, so this is obviously laborious and time-consuming. And then the second is one of accuracy, as with any manual process, it can definitely be error-prone. So QIAGEN has really simplified this process by integrating and centralizing all this information into what's now known, well, the Ingenuity, but really now QIAGEN knowledge base. So for the past 18 years, we've had MD and PhD level scientists manually curating the literature and inputting findings in a very controlled and meticulous way. Now, beyond the literature, we also incorporate data from third-party sources like clinicaltrials.gov, OMIM, and ClinVar together to build this ontology such that we now have this rich resource of gene-gene and gene-phenotype relationships that you'll see in a moment will enable us to easily pull out variants of interest from a clinical exome. I'll point out specific sources within the knowledge base later in the talk, but here, especially on the left-hand side, you'll see that there are several data sources that we've included here in our variant analysis and prioritization tools. But I wanted specifically to point out the recent inclusion of CentoMD, which we're really excited about, as it's the world's largest mutation database for rare diseases. So all these variants are coming from the CentoGene genetic testing service, so all variants are annotated using the same in-house pipeline and associated with the phenotype. So if a variant in your sample that comes through in our tool is present in CentoMD, you'll be able to link out to it through a link, and then you can investigate the allele frequency of your variant of interest in the CentoMD patient population and look at associated phenotypes and pathogenicity. So this is one tremendous resource among, you can see a number of other resources that are really coming together to build the knowledge base and the framework underlying our tools. So we talked a lot conceptually about our content, but let's take a look at the breadth of coverage in terms of numbers. So our knowledge base not only contains two of the most common and frequently and most prevalently used clinical mutation databases with clinical relevance, but you can also appreciate how much value is added from our unique curated content from the literature, as we're adding 121 more genes in addition to what's already in HGMD and ClinVar in our knowledge base. And additionally, in terms of variants, we're increasing just by adding our own curated content, 159% more variants in addition to what's already in ClinVar and HGMD. So you can see the tremendous breadth and depth of coverage that we're offering by including our manually curated content as well as those in ClinVar and HGMD and other sources as well. So to illustrate how extensive our variant coverage and our content is, so Counsel is a health technology company that offers carrier screening and hereditary cancer risk assessment. And last month at the American Society of Human Genetics meeting, Counsel presented their findings in a poster from an evaluation they performed. And the goal of this evaluation was to assess whether utilization of QIAGEN's variant-specific bibliographies within QCI could match the level of accuracy and quality of Counsel's in-house manual curation and pipeline. So they looked at around 2,000 variants that they reviewed over a three-month period. And in the graph on the left, you can appreciate the comprehensive nature of our content, where there's a tremendous amount of overlap between variant-specific publications in the Counsel database as well as that in QIAGEN. As you can see, there aren't very many that are unique to Counsel at all, but you can see the tremendous number of variant article pairs that are added with the use of the QIAGEN knowledge base. In addition to this, you can see that there was a tremendous amount of time saved in looking at variant-specific publications and searching for these publications, which you can really appreciate more fully in the median sort of graph, where the amount of time spent searching for variant-specific publications was decreased by an order of magnitude when using QCI. So overall, Council demonstrated there was clearly a value in adopting QCI Interpret into their variant interpretation pipeline, and they've now made it a key component of their variant assessment pipeline because they felt so strongly that it added tremendous value. So we've talked a lot about the content in the knowledge base and the variant-specific content that underlies QCI-I. So now we'll see the tool in action in a clinical exome use case. So this case was actually published last year in BMC Genomics, and in this trio, the proband has 2 unaffected parents and is exhibiting a constellation of features that are included here. But there was no real clear diagnosis. So to identify the causal variant, whole exome sequencing was performed in all 3 individuals in the kindred. So analysis of the FASTQ files was performed in a separate platform upstream of this and produced a VCF with around 6 million or greater than 6 million variants. Now, to get this number down to something more manageable, we can utilize our variant filtering tool, Ingenuity Variant Analysis, where we filter based on evidence-based implications in the knowledge base. So we filter based on things like call quality, as well as frequency in Nomad and other publicly available databases, as these are also resources that are underlying the knowledge base. And then, as you'll see when we get more into QCI Interpret, we do automatically compute the ACMG, using the ACMG guidelines, we compute and classify pathogenicity. So we can filter based on saying, I only want to keep variants in my list that are pathogenic or likely pathogenic based on those guidelines. And then finally, because we're presuming that this is a de novo event and a de novo mutation in my case or in the proband, we've then filtered to look at variants that are exclusively or independently present in my case samples, so in this row right here, and then absent in my control samples are my 2 parents. So you can see the indicated by these 2 dashes, meaning that particular variant is present in my proband and then absent in my control samples. So now, in just a matter of minutes, using this filtering cascade and the underlying content that we're pulling from these external databases, and also information from the knowledge base, which allows us to impute or to classify variants based on the ACMG guidelines, we've gone from over 6 million variants to just 21 variants, which is something very manageable, which we're able to now export, and we're ready to assess and report out on some of these variants. So using the export function, we can now export this short list of 21 variants over to QCI Interpret for analysis and assessment. So you can see it sort of grayed out in the background. Within QCI-I, you'll fill out some basic pieces of information, including the patient name, the test date, and the SKU for the test being performed. And again, we can leverage the information in the knowledge base to further filter and prioritize variants based on the relationship with a particular diagnosis, if that's appropriate, or a constellation of features which I've typed in here. So these can be HPO IDs. As you can see, you can upload a file with these HPO IDs, or we can just simply type in our constellation of features. So this will apply, and will make more sense in the next slide, a phenotype-driven ranking algorithm of my variants to identify candidate diseases associated with these phenotypes, and then relevant variants in the patient that are associated with those candidate diseases. So now I've uploaded all of my VCF from variant analysis and added the relevant information, so I'm ready to look at my variant assessment. So after applying the phenotype-driven ranking algorithm, we arrive at this list of just 6 variants that you can see here that are ranked based on the likelihood that the candidate disease which is present in this column, is explained by the phenotypes I entered, which are represented. I entered 5 phenotypes, so this Rubinstein-Tavy syndrome type 1 actually it has a connection or relationship with each of the 5 phenotypes that I entered. So we're ranked based on the likelihood that the candidate disease is explained by these phenotypes and has a relationship with a variant from my VCF file. So using this approach, we prioritize this EP300 to be the most likely causal variant with a candidate diagnosis of Rubinstein-Tavy syndrome type 1. And again, it's these genotype-phenotype relationships and the phenotypic ontology that's curated within the knowledge base that enables this kind of prioritization. Without the content used in filtering, we never would have arrived at this strong candidate variant so quickly. And this, again, was all accomplished in a matter of minutes. So I just wanted to point out a few key attributes here, including the automated ACMG classification. So you can see that this variant is classified as pathogenic, and I'll show you in an upcoming slide, exactly what rules were triggered, and we give you and provide all the information to keep things as transparent as possible, precisely how we arrived at this pathogenic classification. And we do this using, again, the information in the knowledge base to apply those guidelines. Again, just reiterating, we also are ranking based on phenotypes, which is a clear, it's not necessary, but it's definitely a clear advantage of the underlying knowledge in the knowledge base that we can rank and prioritize these variants based on these known relationships between variants and phenotypes. And then finally, you can see we can do some further filtering based on, again, the classification. So you see in this short list of 6 that has a connection with my phenotypes that I entered, only one of them is actually predicted to be pathogenic, but we can go through and investigate the others. You can see that there are both pathogenic, which is my one pathogenic variant in my list, as well as a number of variants of uncertain significance. So as I mentioned, we leverage the knowledge base to automatically compute the ACMG classification and show you which rules were triggered, which you can see here, while maintaining the flexibility to add or remove criteria, which you can see here. So we can change the strength of the evidence that we're perceiving, adding criteria, as well as adding notes and adding rationale here as well. And then furthermore, as council has demonstrated the great value, we can also view the bibliography. So this is going to show me variant-specific findings when I click here on whatever particular variant I'm investigating. In this case, I'm investigating this nonsense mutation in EP300. And what we see is there are 20 references for this particular nonsense mutation from the literature, as well as links to OMIM and HGMD, which I haven't shown here, but you can see that these are some citations from the literature, and I can choose to add these to my report by clicking on these little tiles here, so that I can customize and see what information that makes sense for me to see within the report. Another key feature here is I want this to trigger something and learn have I seen this variant before. So we can see prior lab observations where I can say, okay, I've seen this nonsense mutation before and I classified it or assessed it as pathogenic, and this was the particular phenotype and the date that it was signed out on. So you can keep track of the tracking of that variant and how you've assessed it in the past. Additionally, reported case counts. So in other laboratories, for example, in OMIM, in ClinVar, has this variant been seen and what was the significance? Was it seen in an infected individual or an unaffected individual, which can be a rich resource of information when you're really trying to prioritize and understand the potential implications of these variants. So again, with the example cases from other labs, and I mentioned CentoMD, so if there was a citation or this variant was present in that CentoMD patient population, there would be a link here as well. But all of these links go to the HGMD specific accession, ClinVar, as well as Cosmic, or any of these other external databases that we pull data from for you to assess and investigate what the scenario or the context was underlying those variant annotations. And additionally, again, because we have, exact and of course NOMAD, and a number of other population-based data pieces in the knowledge base. We can also look at, frequency of these variants, so the allele frequency in different populations, and substratify actually by ethnicity as well, which is a nice feature if you're looking at a particular ethnicity. And finally, bringing in silico predictors. So we're really in one place, providing you with all the information you would need to assess your variant. So finally, you can see, if I moused over this CAD score, I could see that this variant produces a CAD score of 38, which is quite deleterious. So I can integrate, all these pieces of information to make my final assessment, rather than having to hunt around in several different places. So you can see here that the final output is going to be this report, which has several pieces of information from the patient name and things that we added. It's very straightforward, and this is configurable, so we can work with you to configure this however you like. But it's very straightforward. We show you the reported out pathogenicity. So I stuck with the computed classification that this is a pathogenic variant based on the ACMG guidelines. In the context of this phenotype, this Rubinstein-Taybi syndrome type 1, we have other information regarding the specific protein changes and changes to the cDNA. And then finally, you can see, what in these notes, what version of the knowledge base is being used in all of the different, builds of the different databases and data types that we're pulling from to come together and build a knowledge base. So to keep it consistent and know that when I, reported out this variant, this was the particular, build of dbSNP that was used in the assessment. And also, I selected those certain citations and things that were relevant for me to know to include in this report. So we've got my HGMD accession number, my ClinVar accession number, and a number of citations that are relevant to this particular variant. So within the QIAGEN Knowledgebase within QCI Interpret, it really facilitates two things that I hope I've left you with today. The rapid variant filtering and prioritization, which is essential using our curated content. So we can filter because we have all of these NOMAD and EXACT and NHLBI, ESP, for example, public databases underlying the knowledge base. We can filter based on allele frequency in those databases. We can select for pathogenic and likely pathogenic variants, for example, because we do automatically compute the ACMG guidelines using content from within the knowledge base. And also we can identify causal variants based on relationships with phenotypes and disease indications, because again, we have these curated gene-gene and gene-phenotype relationships that really enable us to uniquely position us to pull out these causal variants based on these disease indications that we're entering. And then beyond that, we also have the other piece is the comprehensive variant reporting, because we do have our automated ACMG variant classification done for you. That you also, we also offer the relaxedness of it, so you can actually add new rationale and add different pieces of information to change the assessment, as well as our variant-specific bibliography. So rather than now hunting around and looking in PubMed in several different places, like Council was able to show, it's going to save a tremendous amount of time and be more, efficient and accurate because of the depth of coverage, the breadth of coverage, and the just like overall content that we're providing here with the variant-specific findings. So that's all I have for you guys today. I'm happy to take any questions if there are any.
Thank you, Dr. Poitras, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window. Type your question into the box that appears on the screen and click the send button. We'll answer as many questions as we have time for. So let's get started. Our first question of the day asks, how often is the content updated?
So that is a very important question. Maybe I can pull up a slide just to give it some context. So I mentioned that there were, let me get rid of that thing. Okay, so I mentioned that there were different sources of information, right? So we've got our expert findings, which are our manually curated from the literature findings, and those are updated on a weekly basis, like pretty much daily. We're always putting in new manually curated findings from the literature. I also mentioned that we bring in a large number of third-party data sources, so these are your ClinVar and clinicaltrials.gov, for example, and those are updated on a quarterly basis. And again, it's pretty easy, especially in QCI Interpret, to see, what builds we're using. Oh, sorry, let me. Of each of those external, you can see the full list of all the external data that we're pulling in the builds for those as well. So again, those are quarterly, and then all of our literature-based content is updated weekly. Thanks.
Thank you. Our next question asks, how is the content curated?
So again, I can go back to that slide. Let me find it. Sorry. Okay. So we do actually have full-time people whose job it is, or at least part-time, to, again, at the MD and PhD level, to actually read and using our controlled vocabulary, put in these expert findings. But we also have these expert assist findings, which uses a computer algorithm to go through and pull out the major findings. So a finding would be kind of a summary of the meat and potatoes of what's in this article, right? Like this gene is associated with a particular phenotype in a particular context. So we have a computer algorithm that does that, but always as a final QC step, actual scientists are looking at those findings before they go in the knowledge base to make sure that we have the highest and best representative content.
Thank you for that answer. Our third question of the day asks, if I have seen a variant in my lab previously, will QCI-I flag that variant?
Yeah, so that's another really important feature to note. Let me see here. An important functionality. So, sorry. So you can see here, so for example, in this EP300 example, I do have this laboratory observations key where it has actually pointed out the assessment when I previously saw it, the date that where I previously saw it, the accession ID, and the phenotype, so I can say or feel more confident that, okay, I reported this out as pathogenic previously in a particular phenotype of interest that may or may not be close to what I'm studying currently, but at least I have that information right in front of me. So yes, it does.
Excellent. Thank you. That was the last question I have for the day. Thank you once again. I would also like to know, Dr. Poitras, if you have any final comments you'd like to leave the audience with today.
I just really hope that I've impressed upon you the fact that our content is really second to none, and will speed up and make your variant reporting more accurate. Additionally, I just wanted to point out that, because I forgot to mention this, that we do integrate with electronic medical records mechanisms, as well as with different limb systems. So we can highly configure this and integrate this with whatever your existing pipeline is to really streamline your variant interpretation and analysis.
Excellent. Thank you so much, Dr. Poitras. I would also like to thank Kaijin for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through February of 2018. You will receive an email from LabRoots letting you know when the webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again here soon at LabRoots. Goodbye.